Okay, so I would like to actually introduce my next speaker. As you can see that we basically, this is speakers that we really need no introduction. Dr. Dale Sandler is the chief of epidemiology branch at the NIHS, and she's going to really enlighten us about some very exciting studies. And I really cannot resist, as she's sitting up here, to let you know that she has some of these most amazing cohort studies, including the agricultural health studies and the sister studies and the two sister studies, and now she's in charge of this Gulf study. So I hope that she will briefly mention some of this in her talk. Thank you.
Thanks. So thanks for inviting me here. Time is short. We're past where we're supposed to be, so I'm going to try to talk fast. I'm also going to talk light. We're going back and forth between epidemiology, excuse me, and biology. So I was asked to talk about lifestyle and environmental factors associated with telomere length. And I'm just going to quickly recap, because you've seen this like 4 or 5 times already, that telomeres, which are non-coding repeat sequences of DNA that cap and are, excuse me. I should have spoken earlier.
Got water coming.
Okay, thanks. But basically telomeres shorten with cell division and DNA damage, and critically short telomeres can lead to cellular senescence, genomic instability, and they have implications for disease risk. And we've also heard that telomeres can lengthen, which also has potential implications for disease risk. Sorry. I don't know. I don't know what happened. I haven't spoken in 4 hours. So as I was thinking this weekend, I had a chance to think about what I wanted to say during this presentation, and had an opportunity to go to a new exhibit at the National Gallery of Art that was called Serial Photography. And thank you. Now I have to. I'll be in good shape. So it's called serial photography, and I was especially struck by a series of photographs of individual body parts of Georgia O'Keeffe taken by Alfred Stieglitz. And the series was described by the artist as illustrating the power of the fragment to express the whole. And so given what we've been hearing, what we've been learning about the many factors that appear to be related to changes in telomere length and the potential implications for telomere length on disease, I thought this was a particularly good analogy in thinking about the potential for telomere length as a biomarker to use in studies of environment and disease. Okay, so we know that telomere length is determined by multiple factors. We've seen data today showing that telomeres on average shorten with age. They shorten about 25 base pairs per year. There are age-related differences in the rate of telomere shortening, and shortening can be accelerated by oxidative stress and inflammation. We also know that there's not a steady decline across the lifespan, which makes it challenging to use telomere length as a biomarker in epidemiologic research. And of course, we've heard about telomerase-related lengthening due to compensation mechanisms. The biologic processes that lead to telomere shortening include mechanisms that are relevant to link environmental exposures to disease, which raises the question of whether or not telomeres function as an intermediary between environmental exposures and disease. In thinking about the role of environment and disease, one important line of evidence comes from twin studies. Twin studies can quantify both the genetic and environmental contribution to diseases or other outcomes. There's one study that David mentioned earlier, actually, of a twin cohort of U.S. male veterans. And his group studied telomere length in 686 male U.S. World War II and Korean War veterans that included pairs of monozygotic and dizygotic twins. I believe that the group was elderly at the time that the telomere length was measured. Although telomere length was correlated in pairs of twins, there was actually no difference in the concordance between the monozygotic and dizygotic twins. And statistical analysis of these data suggested that telomere length in later life was due to shared environment rather than to heritable effects. There's been a growing body of research on telomere length related to environmental factors. Most of this work has been focused on lifestyle environmental factors rather than environment with a capital E. The studies are small, and many of the results haven't been replicated in more than one or two studies. But shorter telomeres have been associated with smoking, with obesity and abdominal fat, and with alcohol consumption, whereas longer telomeres have been reported to be associated with greater physical activity, multivitamin use, dietary omega-6 and omega-3 fatty acids, cereal fiber, and serum vitamin D levels. This is data from an interesting paper on alcohol in telomere length. One of these is a pointer. Is this one a pointer? Okay, so this is a study of older men. Telomere length was measured when these men were on average 76 years of age, but these men had participated in a cohort study called the Helsinki Businessmen Study for many years, and they had information on alcohol use at various time points over the lifespan. And this is not it. Okay. It is it. Where is it? I can't see it. I'm sorry. So in any event, if you look at the top left panel, you can see that telomere length is shorter with increasing numbers of drinks per day. When you're looking at the alcohol consumption 25 years ago, but when looking at alcohol today, there was actually no association with telomere length, suggesting the possibility of long-term damage associated with some external exposure. There have been relatively few studies of other types of environmental toxicants. There's small studies, individual reports, however, linking shorter telomeres to traffic-related air pollutants, to lead levels in Chinese battery workers, shorter telomeres in relation to N-nitrosamines in Swedish rubber workers, and with pH exposures in Polish coke oven workers. On the other hand, longer telomeres have been found in relation to arsenic in drinking water, and in one of these studies, arsenic was associated with increased telomerase expression. Longer telomeres have also been reported in studies of acute exposure to a metal-rich particulate matter in steel workers and in relation to PCBs and other organochlorines. So one of the exposures that's had the most studies, and you can see this is not an extensive body of literature. There are 4 studies looking at various types of air pollutants. The studies are small, ranging from 63 to only 165 participants, and the results are not entirely the same. The differences may relate to the size of the studies, to the specific type of exposure that was being measured, the duration of the exposure, or other population characteristics. So we were interested in the possibility that pesticides might be associated with telomere length, and we were able to do this in a cohort study that I'm involved in. It's a collaboration between the NCI, the EPA, NIOSH, and our institute called the Agricultural Health Study. This is a prospective study of cancer and other health effects of pesticides and different environmental exposure, other agricultural exposures, The study includes 57,000 licensed pesticide applicators from 2 states, Iowa and North Carolina, who were enrolled initially between 1993 and 1997. And we also included 32,000 of the spouses of the private pesticide applicators, who are also known as farmers. We follow this cohort every year through linkage to cancer registries and vital statistics, and we have follow-up telephone interviews with them every 5 years or so, where we update information on their farming practice and on their health. The literature, and especially the agricultural health study, have found links between a wide range of different pesticides and cancer, but the mechanisms linking the pesticides to the cancer are not really clear. Some possibilities include oxidative stress, DNA damage due to the pesticides, chromosome aberrations, immune response abnormalities, and chronic inflammation, which led us to be interested in the possibility that the relationship between these pesticides and cancer risk was because of their influence on telomere length. So we carried out a study within the agricultural health study. Actually, this was part of a case control study where we were looking at many genetic factors in prostate cancer and in controls. So we looked at telomere length in the controls, the 1,300 controls who were chosen for this study. These were cancer-free pesticide applicators, again, from the 2 states, Iowa and North Carolina. We had collected buccal cells from members of the cohort at the second follow-up interview. We collected them from about 40% of the cohort, and we also collected buccal samples separately using a mouthwash approach as people told us that they developed cancer. We did this through the mail. And telomeres were measured with a quantitative PCR assay, and the exposures of interest were self-reported information on use of 48 specific pesticides that we collected from participants when they enrolled in the study. So we found that shorter telomeres were associated with increasing days of pesticide use after taking into account many factors that we considered important to adjust for. We found that there were 4 specific herbicides, including alachlor, metolachlor, trifluran, and 2,4-D, that were associated with shorter telomeres, as well as 2 insecticides that are widely used, permethrin and toxaphene. We had several measures of pesticide exposure, including cumulative days of use of pesticides and an intensity-weighted measure, and the results were similar when we took into account pesticide practices that might moderate a person's exposure to pesticides. And in that analysis, we found the same thing, but also associations with DDT. Many of these pesticides that we found to be associated with telomere shortening have previously been associated with increased risk for several different cancers. We have not completed our analysis for the prostate cancer study, but our preliminary results, unfortunately, do not suggest that shorter telomeres are associated with prostate cancer. So we're also interested in looking at telomere length in the sister study, which is a cohort that I'm leading at the NIEHS. This is a prospective study of 50,884 sisters of women who've had breast cancer. These women are at increased risk of developing breast cancer themselves over time by virtue of having a first-degree relative with breast cancer. The women were recruited between 2004 and 2009 from the United States and Puerto Rico. They were aged 35 to 74 when we recruited them. The key here is that at that time, they did not have breast cancer, although, as I said, they're at increased risk for developing breast cancer, and 16% of our women were minorities. So the basic design of our study is we enrolled these women, and then we collected extensive information from them through a telephone interview and visited them at home, where we collected biologic samples that we banked away. We follow these women every year. We ask them additional questions about changes in their health and exposures. And like women everywhere, some of them develop breast cancer, and people develop heart disease and diabetes and other conditions. And our analyses over time will compare the cases to a random sample of the cohort. I mentioned we collect a lot of data in our study, which allowed us to look at many factors in relation to telomere length, but we're focused on the full lifespan of the woman, where we collect information on exposures and health at various vulnerable time periods, including in the prenatal environment, asking them about their mother's experiences, reproductive years, and so on. So our research on telomeres and in the sister study was actually motivated by some of Alyssa Eppel's papers. A fellow in our group, Christine Parks, is very interested in stress and immune response, and she got a grant to look at perceived stress and telomere length. So it was our first look. This is a cross-sectional study where we used baseline data collected from women who joined the sister study. We had questionnaire data on self-perceived stress and other factors. We measured height and weight, as I mentioned, as part of the clinical exam. In this study, we measured stress hormone levels in urine samples that had been collected at baseline, and we measured telomere length in leukocytes. This is a small pilot study involving early participants. There are only just about 650 women who participated in the study. So again, we used a quantitative PCR assay to estimate telomere length. We used DNA from leukocytes, actually from frozen whole blood. This assay looks at the ratio of relative amplification of telomere sequence to a single copy gene. And in our study, we included, for quality control purposes, duplicates, and the coefficient of variation was reasonably good. And we adjusted for plate effects, which are something that seems to plague our assays. The average telomere TS ratio values in our group, this study, were normally distributed with a mean of about 1.32. And for purposes of reporting, we transformed these results into base pairs so we can look at base pair differences. Too many things. So here are some of our early results thinking about stress and telomere length. When we look at perceived stress, it's important to keep in mind that we had very low levels of perceived stress in this cohort. This is a cohort of women who volunteered. They're concerned about their health because they have a sister with breast cancer, and they took charge by joining a study. So the stress levels were low. But even so, we do see that telomere length is shorter for women who reported higher levels of perceived stress. And when we looked specifically at women who had a biologic stress response, and here I'm showing women with higher norepinephrine levels, you can see that the greater the score on this standardized measure of perceived stress, the shorter the telomeres. We also looked at characteristics of work life as a measure of stress, and we found that women who work full-time or overtime had shorter telomeres. We also found similar effects for women who had night work and shift work. And in both of these analyses, the effects were greater among women who were older, women who reported other stressful events in their lives. Finally, we were also looking at stress and started to look at stress in childhood. And in one of our studies, we were looking at multiple measures of childhood adversity, including having a very young mother, having parents with low education level, or having periods of time in your life when you weren't getting enough to eat. And we see that telomere length decreases with increasing numbers of these adverse stressful events. In another analysis using these same data, we looked at obesity and telomere length. We found that women, looking at body mass index today, we found that overweight and obese women have shorter telomeres than women who were of normal body mass. And we also saw a similar association for women who were overweight in their 30s. when we combined the time periods, we found that the greatest effect was seen for women who were overweight and for their entire lives. Now, I thought this was sort of a too good to be true story. When we also looked at the use, we actually we were looking at antioxidants. Our concern was, our interest was in specific antioxidant vitamins, and we found no association between the use of antioxidants and telomere length. But we did see that women who took a multivitamin every day actually had longer telomeres than women who did not. I can't claim to say that this has anything to do with telomere biology, and they just reflect characteristics of the women who take vitamins on a daily basis. So we found a number of other things that were related with shorter telomeres in this analysis. We confirmed decreasing telomere length with age. In our study, about 27 base pairs per year, which is consistent with other reports. We found shorter telomeres in current smokers, those with lower education levels, women who reported that they were in poor health, and also in women with cardiovascular or metabolic disease by self-report. We looked at other factors that weren't related to telomere length, including clinical depression, the average sleep duration, regular exercise, and use of hormone replacement therapies. So in thinking about the role of environment in disease and its relationship to telomeres, what we're hoping to see is that environment, lifestyle, and stress may, if they have an impact on telomere length, that's the telomere length then will lead to health effects. So we're looking for telomere length as an explanation if we're thinking about the full story. But it's also likely that these same factors may be associated with telomere length changes and also separately with disease. And so in which case, this is just sort of a general marker of susceptibility rather than a causal pathway. So in that regard, we were also interested in telomere length in relation to breast cancer. This is a separate sample of the sister study cohort. We were interested in breast cancer because of the case control studies showing that telomere length is shorter in tumor tissue from breast cancer patients, and also in cases in the blood of cases with breast cancer compared to non-cases, and also because we'd seen many of these lifestyle factors that are related to breast cancer risk were associated with shorter telomeres. And the short story is that we saw absolutely nothing. There was no relationship between telomere length and breast cancer risk. And it didn't matter if we were looking at cases that were diagnosed shortly after the blood draw and those that were diagnosed longer. Of potential interest to the rest of this conference, in a preliminary analysis, we do see some evidence that epigenetic regulation may be involved in telomere length. So there are some limitations of all of these studies, including ours. The sample sizes are small. The results are inconsistent and unconfirmed in other studies. Results could be due to differences in study designs and other population factors. There's limited data on the reproducibility and validity of the assays that we're using in large-scale studies. There's some evidence that the reproducibility is not bad, but there are other laboratory concerns. More importantly, most of this research has been cross-sectional. We know that telomere length and the rate of attrition vary across the lifespan. There are heritable differences in telomere length that could affect the rate of change, and a single measurement at one point in time may misclassify telomere characteristics. And so we certainly need to see longitudinal studies if we're really going to make sense of this, which brings me back to this exhibit at the National Gallery, which I highly recommend if you have time before your flights on Friday. And this is from a series of photos, actually one of my favorite series of photos, where Nicholas Nixon photographed his sister and his three sisters in 1975. And he continued to photograph them in the same order every single year. So here are the sisters in 2011, and you can see a lot has happened between 1975 and 2011. They've certainly gotten older, but it's not just age. So as part of this series, this is just sort of a frame of many of the photos. If you can look closely, you can see obvious differences in their clothing, their hairstyles, their weight. You can see pregnancies occurring. And you can also see differences in the relationships among the sisters over time. So in the very first picture, they're all sort of standing separately. One's a teenager, and why are you doing this? And at the end, you see that sort of, the close bond between the sisters. So I think for me, this sort of captures the essence of what we're trying to do here. We're trying to have a single measure that's capturing all these many different factors that affect telomere length, which may in turn affect disease in a single measure, and it's quite a challenge. So these are just some of the collaborators on the telomere work and the sister study and the agricultural health study. I finished in time.
I think we should take one question, then we can enter into this general discussion. That would be great. Oh, maybe two. Okay, so given the red light for two.
Helmut Zarbel. So you gave a long list of factors that were associated with shorter tumor lengths. Are they additive if you look in the same population?
Well, the results that I've shown are adjusting for each other. So those were individual effects. And so I don't know whether I didn't actually look to see if you can add them up.
That might speak to dose response at some.
Yeah, that would be interesting to look at. We did see dose response for the psychosocial effects, but haven't considered those other lifestyle factors. I also should point out that the telomere length changes that we see in some of these factors are comparable to the changes that you see with, big age-related effects. And so to some extent, they seem unbelievable to me, but we've seen them and other people have seen them, and so it's intriguing to think about.
Sarah Gerald, USGS. This is a question for you, but also for the panel at large. I was very intrigued by the responses to different stressors, but even more intrigued by the childhood shortening in telomere length. And it made me wonder about various stressors that we and other animals go through in the course of our life as a normal part of our life. Scientists at USGS work with birds and fish. Birds and fish migrate. They fledge. They leave the nest in peril. They go through all sorts of environmental stressors that are just part of what they do. Insects pupate. There are salmon in the West Coast that, as they go upstream to spawn, they fall apart. Basically, their bodies fall apart. And I'm wondering if anyone has done any work on these species in the course of their normal lifespans, their normal life histories.
Somebody else want to answer that question?
Okay, we can take that question when we have a general discussion.
I'm Babs with NIEHS. Thank you for a wonderful series of 3 talks. And in listening to them and putting it together with Thea's talk in the morning, I'm struck with a problem that is both a question from an epidemiological study standpoint, but also a broader question, which is we're seeing tremendous heterogeneity over time from the stochastic effect, tremendous heterogeneity within one cell type, within one person, and then across cell types. And so anytime that we're looking at an epidemiological study, we have to be asking how many cells from that person are you looking at? Where are those cells coming from? And in the sister study, you're getting buccal samples, but you're also getting blood samples?
No, the sister study is blood. The agricultural health
study was buccal. Okay. But in any event, you're kind of getting them at one point in time, and they're one particular tissue. They're never the tissue of interest. And so are you thinking of ways to maybe, from just your study standpoint, ways to tie back collect a specific sample or somehow supplement the study to get at that issue?
So in terms of the one sample issue, one of the things that we're planning to do is to collect additional blood samples from the participants. We actually have tumor tissue, but it's in tumor blocks from the participants. And it's, of course, you only have that from the women who get breast cancer because women don't like to give you a breast biopsy either if they don't have to. It's not just men who are squeamish about surgical procedures. But we plan to go back and take a random sample of the cohort and collect another blood sample where we can start to look at changes over time. One of the concepts that comes up in with the psychosocial stressors that really wasn't discussed but is relevant in my life in the Gulf study where we're studying oil spill workers is the concept of resiliency. And so faced with the same stressor, not everybody responds the same way. And some people have really terrific coping mechanisms and some people are energized by stress, like probably everybody in this room who was, up late last night fixing their talks. That's what keeps us going, and hopefully, work-related stress isn't a problem for us.
And related to that, and a question for the panel is, when you have processes that can both shorten and lengthen, in different cells, and then you're taking a population, how do you address, the summation of those two processes?
I also want to say, I think in looking at some of these studies more closely, the shortening and the lengthening, that was actually sort of disturbing to me, that you can have long telomeres and get cancer, and you can have short telomeres and get cancer. And so it seems like you can look at this and you can create any story that you want if you're an epidemiologist. But looking at the specific occupational studies that have been done, it does look like you first get the long telomeres and then over time they get shorter. So maybe it makes sense, but I'm not a biologist.